Want to get caught up on the latest, greatest bioprocessing equipment and solutions without putting in travel requests, flying across the country, spending gobs of money and time getting harassed by salespeople? Of course you do. Register for Bioprocess Online's Bio Expo Live, being held July 30th through August 1st. This inaugural expo is a fantastic opportunity for biopharma companies and contract manufacturers to evaluate the latest and greatest from the comfort of your desktop or mobile device. Conveniently, we've broken down the lineup into Upstream Solutions on July 30th, Downstream Solutions July 31st, and Quality Analytical and Data Solutions on August 1st. It's absolutely free to register for this series of short, digestible, and interactive sessions. Just hit the link in today's show notes to register for Bio Expo Live today. From the outside looking in, drug discovery might look like a bunch of scientists throwing spaghetti at a wall to see what sticks. And while it's a crude way to put it, and in fact demeaning of the important work that scientists do, it's not an entirely naive assumption. Disease pathology is incredibly complex work because disease progression is influenced by myriad biological, chemical, physical, and environmental factors beginning at the molecular level, like DNA and the RNA it transcribes to, the polypeptides RNAs code, and the proteins RNAs become. This complexity of disease pathology is only exacerbated as those proteins reach the cellular level, where any permutation of chemical, physical, and environmental factors influence disease progression. Rudimentarily, finding the silver bullet that will stop disease in its tracks or prevent it from starting in the first place means finding the influence, or more appropriately, the permutation of influences of disease and altering their course. So yeah, sometimes that looks like throwing spaghetti at the wall. I'm not telling you anything you don't already know, but that's another big wrinkle in the paradigm. There is, in fact, a lot that we don't already know, and the more we discover, the better our odds of getting sticky spaghetti on the wall. For instance, for many decades, it was generally accepted that just 2% of the 3 billion base pairs that comprise human genomic DNA represent protein coding sequences. Only recently have we discovered that, in fact, 80% of DNA is transcribed into RNA. If you're a molecular level drug discoverer, that's a potential game changer. Dr. John Lepore is a molecular level drug discoverer, and his company, in further exploration of our new understanding of DNA transcription, just might be on to some sticky spaghetti. Founded in 2020, the flagship pioneering startup ProFound Therapeutics began with a simple question. What if more RNAs were being translated into proteins? Answering that question took ProFound down the rabbit hole of the translatome, where it's now studying the full compendium of RNA sequences that are being translated into proteins. Along the way, the young company is gaining confidence that its research will reveal important insight into potential therapeutic protein targets and medicines. I'm Matt Pillar. This is the Business of Biotech, and on today's show, I am thrilled to bring you Dr. John Lepore, CEO at ProFound Therapeutics. He hasn't always been a drug discoverer, but we're about to learn how he got there and where he's taking ProFound. Dr. Lepore, welcome to the show.
Great, Matt. Yeah, thanks to be here, and a wonderful introduction. I'm going to steal pieces of that liberally because it connects many dots together. And you're spot on that it seems like a somewhat stochastic or random process. But it's really more. I would frame it up as the risks are high up front, and the whole process is to try to minimize risk as you go. And we call it risk discharge, to get to something that you're more confident in, to get to patients. And all the things that my company are doing and many other companies are trying to figure out how to streamline that process and make it more likely to get to the end. But thrilled to have the opportunity to talk with you about, I guess, how I got here, but also what we're doing.
Yeah, we're going to start with how you got here, because when I look at, like, you were a physician scientist, when I look at your career, I my first thought was like, this guy's been swimming upstream his entire career. And I don't mean upstream as in like against the tide or against the current. I mean upstream in terms of from the patient as the whole, to, as I said, drug discovery at the molecular level. And I can kind of rationalize that. I don't know if it's the best analogy, but I can kind of rationalize that by just giving a brief synopsis of where you've been. You earned your MD at Harvard, practiced medicine at Mass General for 7 years, and taught at Harvard for 3 years, also serving on Penn's faculty for 6. So like that, to me, like I look at that and I'm like, okay, well, there's the academic and physician scientist part of the, continuum. But then you joined pharma with GSK. You stayed in R&D roles there for like 16 or so years, which is quite a long stint, culminating with 5 years as SVP and head of R&D. So now we're squarely in the discovery phase of your career. And then last year you joined Flagship, became an entrepreneur in an even further upstream sort of capacity from my perspective than typical R&D at ProFound. So I'd like you to, rather than me just kind of like make assumptions,
right?
Walk us through that arc and the why behind the decisions you made, starting with why you decided to stop practicing and teaching and make the move into big bio, as it were, with GSK.
Yeah, no, thanks for introducing the background, and happy to walk you through some points. For me, it's always been about what's the way. I'm really driven to figure out how to apply science to help patients. And, life's pretty short and there's not that many opportunities. And what I've done in my career is try to find the places where I could apply my specific skills, intellect, interest, energy to have the biggest effect. And, I absolutely love seeing patients, and I have enormous respect for the people who do it full time. It's extremely hard work, physically, mentally, obviously extremely important for society. But for me, I was just so compelled by understanding the scientific underpinning of disease, and you described it well at the in the preface so well about how
it's but I mean, obviously, painting with some broad brushstrokes. I mean, the influences, like when you're a physician and you're practicing and you're using medicines and therapies that are provided by industry, there's required certainly an understanding of disease pathology,
a deep understanding. And to do it well. And even in the 20 years or so that have passed since I really practiced medicine, it's becoming more complex. And that's why you see hyper specialization, where people only take care of one particular subset of disease. For example, gastroenterologists that really specialize in inflammatory bowel disease, because there's so many different therapies that you need to know the molecular basis of how they interact with the patient, but also, which particular subtypes to give them to and in what order, what side effects to watch for. So it is a complex interchange of science and medicine. But for me, getting back to how I made the decision to switch, I was really enthralled with how can I be involved in creating one of those entities that has such a profound effect on patients. And the arc that you've described of my career was a series of decisions that I thought helped position me best to be able to do that. Mm-hmm. And maybe the last thing I'd say on not seeing patients, I still miss it. And you could ask the patients, but I think I was reasonably good at it. I miss the personal interaction and sort of seeing the outcome and people feel better, get better, but you can't do everything. And I think I've successfully steered in a direction where I could help many more patients through, the companies I've worked with and projects we worked on.
Yeah. What were you, you mentioned that as a physician, the deeper your understanding of disease pathology and disease targeting and the way that therapies work, the better. What were you feeling confident about? What did you feel prepared for when you joined industry, and where did you, and on the flip side of that, where did you feel like perhaps even with that rich physician's experience, where did you feel like perhaps you were lacking?
Yeah, so the drug discovery process is complicated and specific and detailed and hard. And I think even as a physician scientist, I didn't understand the depth of experience and specialization required for each step. So, it's simplistic to say, well, okay, we'll pick a target, we'll make a small molecule of antibody against it, and then we'll test it in patients. Well, going from the make a small molecule to test in patients is like 1,000 steps of people who are expert on, pharmacology and product supply and product development and how to design the phase 1 and safety testing. There's like 50 different things. Mm-hmm. So that was one thing that was an eye opener for me. It was just the complexity and the depth of like specialization required to go from step 1 to step 2.
What was required of you to make that adjustment, like to embrace that?
That's a really good question. And so you mentioned, why did I step away from seeing patients and teaching? And I just did challenge on that point. I didn't really, because what I learned a lot about, not so much from seeing patients, but from teaching is that, to run a good department and to develop people, I still continue to teach. And I even do that here in ProFound. We could come up on that in a minute. But what I did is become a student in that case. So I was fairly accomplished at that point in time, was, fairly on in my career, had a fairly senior role. But I decided like I needed to like do all the basic steps, like fill out the blank document for the regulatory agency, go to the meeting where we're just figuring out, how many pills to make and, what substance goes in them. And I just did all the basic stuff for about a year or two. And then I felt like, okay, now I know the nuts and bolts of how this works. And that stood me really well. And I've always encouraged all the people I hire and train you've got to take 3 steps back and do the hard work, and then you'll be trained up to do it.
Yeah. During your time at GSK, like I said, you were there for quite a while, and that stint culminated with a pretty high-ranking position there. What did you find was most rewarding? I mean, you mentioned that like part of your motivation to make that transition was to broaden the impact, right? That you could have on a patient base. What was that sort of the most rewarding thing, or were there other like, John Lepore specific rewards that you
I'd say
came out of that?
Yeah, I'd say that's the sort of macro one that cuts across. But then there's like, specific accomplishments, victories, challenges, whatever that you feel happy about because they enabled the macro one. And the macro one for me always has been, always will be, like, how do I have the biggest effect on patients through what we do? Because why waste time on something that's not going to have the biggest effect? But then there's things you need to tackle to get there. And, one of it's the best thing and the worst thing about big pharma is it's a huge organization with deep expertise and people who know things in just great detail. But it's also big and complicated and hard to maneuver. Mm-hmm. And, I think one of the things I was particularly fond of and I think helped a lot of people through is how do you maneuver in that system and make sure things get from point A to point B quickly so that they could then get to patients most quickly. So that sort of learning organizational dynamics and just how to make things get done was an important part of it.
Yeah. Yeah. So fast forward to last year, in fact, you made the decision to leave GSK and join a startup, which is, you talked about risk earlier. Like that's personally like that's a pretty big risk. What was the motivation there?
Yeah, look, same thing. So first off, I was going to challenge the notion that it was risky. Because risk is specific. So for me as an individual, it wasn't so big of a risk because of the background I've had, and I felt like really capable and empowered to deliver on what needs to be done here. And it was an opportunity that sort of uniquely fit my skill set. So it was a great match. So in that sense, it was less risky. But I get the point that it's going from having thousands of people reporting to me to now like dozens. And that's
that. And I mean, thousands, there's that like daily. And then there's like, I mean, obviously you would reach the point in your career where you had achieved success and you probably had some level of comfort in terms of like career trajectory. But there is, at GSK, 16 years there, there's a certain degree of comfort and confidence, right?
No doubt. Yeah. And hard to walk away from for sure.
Yeah.
But the scientific opportunity that I saw at ProFound was just outstanding and unique. And when you get to the level I was at, you get a call almost every week. Hey, I got this company. I got this. Can you come run our blank? So I was getting calls all the time.
Mm-hmm.
But this one had this unique feature, and you introduced this a little bit in the beginning of the call, of opening up a brand new area of science that essentially no one has been. There's been little snippets out in the literature about, but we really explored what's the size of this opportunity of novel proteins. And the data I saw as I was looking at the job told me, wow, like there's enough here to be convinced that this is going to be a very huge opportunity, not for me, but for patients and for science, and to figure out how to unlock this systematically. And it just seemed like a fabulous career challenge, and also fabulous opportunity to help patients even better than I could have staying in large pharma. So here I am 8 months later.
Yeah, very good. And we're going to get into the details around that giant opportunity here in just a minute. But oftentimes when I have conversations with folks who have worked in big bio and big pharma and made the transition, we talk a lot about moving from a super well-resourced organization to moving to an organization that's just by design has to be more agile, more nimble, more frugal. Right? Maybe doesn't have quite as many people to turn to when you've got questions.
Yeah.
Excuse me. So what? Tell me a little bit about that. Like, what did the GSK experience prepare you well for in your role at ProFound? And then on the flip side of that, like making that transition, what did you feel perhaps a little bit like uncomfortable with the adjustment? Right? Maybe you had some some discomfort around the lack of resources, for instance. Like, what were the sort of juxtapose those experiences and tell me what was great and what was maybe not as great?
Yeah. So what prepared me well was definitely having just a breadth of experience. Because in a small company, and particularly one where we're working in a novel field and it's across many different therapeutic areas, every day there's like a completely unanticipated question that comes up. It might be a scientific one. It might be a practical one about how to make the protein or something, or it might be one that's more related to how the medicine would look 15 years from now when it comes. So there's a new question every day. So having spent 16 years in various parts of R&D really helped me prepare to see the questions, like look around corners and anticipate what questions are going to come up, and then also to know where to look to get them answered. So that's was great preparation. The other thing I'd say is I did a lot of business development at GSK. I ran the BD department for a year, and then we also did a lot of deals. And so I was on the other side of the table working with biotech. And that really helps me now to think about, well, how are pharma thinking about my company? And what are the questions and challenges that I'll need to be prepared for? So I'd say those two things. In terms of what was a surprise or a challenge, intellectually, I knew it was going to be a much smaller resourced, much less resourced group in terms of people and money and all that. But you don't understand that till you actually feel it. It's like, it's true. And, a question comes up and I just subconsciously think, okay, I know exactly who in GSK spent 15 years doing that thing. And I could call her tomorrow. It's like, okay, wait a minute, she's not here anymore. So now what do we do? So that was a little bit of a challenge. Like, okay, I've got to create a new network and a new process for getting things answered. So there's been challenges and opportunities.
Yeah. You mentioned BD, your time working in BD at GSK. And I mean, I know as well as everyone else that BD is integral to leadership of a biotech, right? Like that BD experience is, if you don't have it, like you better get it quick, right?
Yep.
So
it's the bread and butter.
What's that?
It's the bread and butter of the whole thing.
It's the bread and butter, yeah. And I want to get your perspective on how a physician scientist, an academician, like someone like you who comes out of Harvard and then spends that time in industry, even during your time at GSK, like when you took on that BD role, and then the further evolution of that from GSK to absolute leadership of a biotech, the skills that you needed to acquire to be a businessman in this space. How did you do it? Like, how did you prepare for that BD role at GSK? And then how have you prepared for, sort of taking on the whole enchilada, for lack of a better term, as the leader at ProFound?
Yeah. So the one at GSK was, I guess, almost accidental in the sense that there was an opening between the person who was leaving, and then there was a leadership transition. So I was asked to do it on an interim basis, which sounded like a couple of weeks just to keep things steady. It ended up being a whole year and many deals and great opportunities. So I didn't prepare for it. It just was sort of there. But, I'd say the things that enabled me to take that on are, first of all, having done deals before that, just as participating in them. But then, BD, just like drug development, ultimately, it's about clarity on how the science relates to an ultimate application of the science to a patient for a specific value proposition. And once you really understand that and could articulate that and know the vocabulary and know the questions and know the risks and challenges to get there, then BD becomes, okay, it's the deal and the terms and the math around it. Mm-hmm. So that's what I brought to the table. It's just a real deep understanding of it's not a scientific question, it's a business question, how the science translates into benefit for a patient. And then that's what could be sold to, people who buy it. Yeah. So understanding that well is what helped me with BD. And it's the same thing I need to do now. It's, all this science is great, terrific, but I'm not here to sort of do human genome 2.0 or write 10 Nature papers. That's a different job. Mm-hmm. I'm here to help the science get visibility, get uncovered, but then rapidly translate it to medicines.
There are plenty of physician scientists who attempt that role, attempt to embrace it, and find very quickly that they don't like it. It's uncomfortable. It's a skill set that they're, like it. I've had conversations like this before where I equate it to, like, if I suddenly decided I want to go work in our sales department, like I don't think I would do well. You know what I mean? Did you have a level of discomfort that you needed to wrestle to the ground with that, or was sort of that pragmatic assessment of the job enough for you to embrace it and be comfortable in that role?
Yeah. The BD, in case any of my BD colleagues are watching, I don't mean to undermine it and say it's all about just the terms and the numbers. I mean, there's so many elements.
Not at all. No, I have profound respect for, I just said profound, didn't I?
Yeah, we'll get to the word profound in a minute. Yeah, because there's a very purposeful choice of that word. But anyway, but yeah, there's so many other elements, both for working in BD, but also for being a CEO of a company. And some of them are just, they are pragmatic, like how to function and bring people together and visualize what next steps could look like and bring people along on an intellectual journey where they say, wow, okay, now I see where you're going. I'll give you money. Mm-hmm. That's not, it's a skill.
Yeah, very good. All right. So I want to talk about ProFound, and I want to learn about this expanded proteome concept. You joined the company last year. The company was founded in 2020. What was sort of you mentioned already that you walked in and you saw an opportunity in the science and in the discovery that you thought would be huge. What exactly was that? Like, what was it that flipped the switch in you, the premise of ProFound that flipped the switch in your brain that said, this is what I'm going after?
Yeah, sure. So first off, the name, I didn't come up with it. I inherited it, but I love it. And first I thought it was a little bit presumptuous, but it isn't intended to be at all. It's really to signal we're finding proteins, ProFound, with an intent to have a profound impact on patients. And it kind of encapsulates the whole thing for my team. And, that's one of the things that I brought to the company is really talking about the transition from pure platform company. And we're still a platform company, but it was a pure platform company when the platform was being created. But now we're in transition from a platform company to a platform company and a pipeline company. Mm-hmm. And, my job is to really make sure both things thrive. I'll tell you more about the platform, but also to make sure there's visible, tangible products coming out that everyone sees as valuable to patients and therefore valuable as a business. And, the first, I'd say, recognition of that was the deal we recently did with Pfizer, which we could talk about later. So, they're the first to really recognize that, and there will be others, I'm sure. But what we do is, as you introduced at the beginning, we started with the premise that there's 20,000 known proteins from the Human Genome Project about 2 decades ago. But when they did that project, and with enormous respect, and that was such an important landmark in science, they necessarily need to have some rules where they defined what a protein needs to be included in their list. And they were restrictive rules by design. It turns out if you relax the rules a little bit, then you start to say, well, wait a minute, there might be other types of proteins. And as you alluded to, there are many types of RNA that we thought, there's one class called long non-coding RNA, meaning it doesn't code a protein. It turns out that it looks like probably hundreds or thousands of them actually encode a protein, which is what we've defined. And the way we did it was we developed a particular technology. It's called RiboSeq. I'll just spare the details, but it's a way of identifying proteins in the process of being translated. It's therefore unbiased. And we find all the 20,000 known, but also everything that's now newly described. And there's literally tens of thousands of other ones. And what we have is a platform that enables us to rapidly, through a database, attribute sort of in silico potential function to them. And then we have a platform to do experimental assessment to see what they really do. And we have a heavy dose of looking at which ones have human genetic evidence for being causal in disease. That's one of the things I brought to the table, is because at GSK, I was leading on the collaboration we had with 23andMe. So, this approach of using genetics to really pick the most promising targets is one that I've utilized here as well. So look, it's no longer a theoretical proposition. We've identified quite a number now of novel proteins that have drug-like properties, where the protein itself could be a drug, or drug target-like properties, where we're making antibodies against them, for example.
Super exciting, yeah. When I talk with the leaders of platform companies, I've often had the conversation around whether the pipeline development is sort of like a floor model, or whether there's intent beyond demonstration. What's the take at ProFound?
Excuse me. Do you mean a floor model in the sense?
Floor model is in the sense that, like, a company will create a platform, and they the platform is onto something. It can make important discoveries. It's capable. The technology is capable of moving the needle, right? Moving the ball downfield. But we don't necessarily have we theoretically don't necessarily have the intent to take the ball any further than discovery. Let someone else develop a pipeline. We'll be happy to sell our platform as a service or to partner. And oftentimes, like I've had conversations where it's been put to me point blank. We did that. We couldn't get anybody to get on board, so we said, well, let's demonstrate it via a pipeline. A candidate. Develop a candidate.
Got it. Sure.
To prove the theory. But the intent isn't necessarily to say, well, we're going to have a follow-on candidate. We're going to have a candidate beyond that. We're going to build an entire pipeline. So I'm looking for like what the sense is at ProFound. You said that your role right now is kind of two-part in terms of the platform product and also developing the pipeline. Would ProFound, and maybe it's too early to say, and I don't mean to put you on the spot, but would ProFound like to develop a pipeline?
Yeah, let me put it this way. The science is applicable across every therapeutic indication, every therapeutic area. And at the moment, we focus predominantly on oncology, immunology, and metabolic disease, but it's equally applicable across the others. And there's no way any single company could prosecute all of that.
Sure.
So, for example, there's no company who could take all the 20,000 proteins. There's like many dozens of companies working on that space. Yeah. So it's not, we won't be a company that owns it all, takes it all, never partners anything. We've already partnered with Pfizer. So, I think to maximize opportunity for patients, we're going to have to do some combination of internal activities, partnerships with the right people. And that's where the BD thing comes in. It's a matter of, for each area and even each asset, what's the most efficient way to get it to patients, which almost always means everybody makes money also, and it's a good business model. So we're going to be exploring and implementing different business models across that space. But, it is early and we have optionality across all of it. But yeah, that's what's fun about this. There's going to be lots of great press releases for you to follow over
Sure.
time.
Yeah. Yeah. And I mean, my opinion is being formed along the way. Early on, I thought, well, I don't know how much I want to, how much focus I want to put on pure platform companies, because I'm the intent of the podcast is to talk with companies that are developing therapeutics. As sort of to your point, it's exciting. The optionality is there. And as I learn more about this and see the companies that are coming on board and perhaps going to build a pipeline, maybe not, maybe just going to partner, maybe a combination of all of the above. Maybe it doesn't matter so much, right? Like the science is cool.
Yeah, and sometimes you can't know what you're going to find in the platform. I mean, to exaggerate for effect, there's like a range of what platforms can do. The worst case scenario is you spend 4 or 5 years making a platform that's really only enabled to make one asset. Mm-hmm. Well, that's not exactly probably the best use of capital. That's just like some amazing asset. But then it's not really a platform. It's more of that was the research to get to the asset. Yeah. The other extreme, I think, is where we are, where the platform has been successful. You identify this whole new area of science, and now you've got like dozens and dozens of target and drug opportunities. And again, that's where I think we're going to necessarily need a mixed model of some sort and variable sort. And yeah, it'll be fun.
To just look at this point blank without the benefit of conversation and learning, to look at what ProFound is looking to do and what it's discovered and what it's looking to capitalize on. It's easy to overstate, right? I think from my perspective, it would be easy to overstate, like, wow, there's a whole new world of proteins that have yet to be prosecuted, yet to be, researched and applied. That's it's revolutionary. It's, pick your adjective, right?
Right.
It's easy to overstate. So I want some perspective on that, some pragmatic perspective. Like how do you characterize this opportunity in terms of sort of historical reference points? Is it comparable to the quote unquote discovery of X or the development of?
Look, at the risk of going down the same track that you're describing of overstating it, let me just frame it this way. For the 20,000 known proteins, and I've seen different numbers, but they sort of center somewhere around 400 or so of the proteins have become drug targets. So a small fraction. If from these tens of thousands of proteins, there could be even a quarter of that or half of that, that's fundamentally important for patients. Yeah. And let's just leave it at that. The opportunity is very, very big. Is it, as big as the genome project of this or that? I'll be extremely happy when there's dozens of medicines that come out of this.
Yeah. Yeah. How are you, like, how do you layer in, we're going to talk about the Pfizer deal and sort of where that lays in terms of the therapeutic discipline of that deal. But how have you navigated sort of the disciplines you mentioned earlier, immunology and oncology and metabolic disease? I guess the question would be how and when do you determine, like, hey, what we're discovering has applicability in these specific disciplines and why?
Yeah. So we set out to work in these three disciplines, oncology, immunology, and metabolism, with a very specific approach and strategy in mind. First off is in oncology, you'll be aware of the rising importance of the class called antibody drug conjugates, or ADCs,
Mm-hmm.
that have been really revolutionary for oncology patients. And there it's an antibody that's engineered with a toxin on it to kill cancer cells, but based on there being a cancer-specific antigen. So the ADC could kill the cancer cell, but not normal healthy tissue.
Mm-hmm.
Revolutionary, HER2 and others. But there's only about 10 targets in that whole field. And almost every day you see a press release of somebody buying one for 100 million or a billion, depending what phase. And what companies are doing now, they're doing their best to find additional targets. But that's been very, very hard. And what there is a lot of momentum on is iterating on the modality, on the antibody, like
Mm-hmm.
the next generation. What we're bringing to the table in oncology is a new set of antigens, of targets. And the way we did this is by identifying novel proteins that as I've described.
Yeah.
And we found quite a few that are expressed on cancer and not healthy. And they're ones that no one else has seen before. And we're starting antibody drug discovery programs and have ADC programs in flight. And it's very exciting because, if for a field where there's only 10 or so, adding 2, 3, or 4 more is just phenomenal.
Yeah.
So that's why we started there. And then the other thing we're doing is finding in immunology, as you know, many of the drugs target circulating cytokines or their receptors. And they're definitely not easy, but they're relatively easy compared with small molecules, which are, you need tons of chemists and it's hard work to make the molecule. And, you've got tissue penetration issues, et cetera, PK, whereas making an antibody cytokine is relatively straightforward. And we've identified through our platform, I mentioned very briefly the database, but once we identify these small proteins, we know tons about them. For example, we know if they have a signal peptide and therefore look like they would be secreted. So we could very quickly figure out which ones are produced by T or B cells, have a signal peptide, and have a genetic association with human disease. That's like fabulous as a starting point for a drug discovery program. And that's what we're doing in immunology.
Yeah. Yeah. Very cool. So, the modality, that you'll choose is not, that's not necessarily predetermined or biased going in.
We've biased our selection of targets to those that are amenable to an antibody.
Mm-hmm.
There's many of these that are intracellular, and that might be targets for small molecules, oligos, degraders, that we're putting less emphasis on at the moment, but there's many of them. And that's the kind of thing where partnerships might be really important.
Yeah. Yeah. And the part I want to get back to a question around the BD and the partnership approach in the context of this, this discovery, this new discovery that hasn't necessarily, to your point, that the translatome is in bits and pieces in the literature, but it's not like you don't go to JP Morgan, for instance, and start talking translatome and just seeing heads nod in all of your your meetings. You're just not going to see that. Yeah. It's new, it's new. So, when a new approach to developing therapeutics is presented, the risk in terms of partnership and funding is sort of ratcheted up a notch. And you've said, it's BD's tough. It's a hard business. How is ProFound, and in the context of ProFound's relationship with Flagship, how are you uniquely situated to embrace the risk of pursuing science that some in the traditional space might consider a bit too soon or early for significant investment?
Yeah, there's a lot there, but maybe I could give you some.
No, that was a very,
how we think about it.
Wordy. It was a very wordy question.
No, yeah, maybe, but there's just a lot of elements there that I could sort of tease out. Let me see if I could give some structure to it. First off, in terms of the interactions with pharma, It all depends on getting to the right people.
Mm.
Because if you get the right scientists who understand what we're doing, that you usually see the light bulbs go up like, oh, that really? Wow. Okay, tell me more. And sometimes it takes, a second conversation where the first group is like, well, that's interesting. I don't know. Then they talk with their colleagues and they come back and they say, oh yeah, okay, now we want to see that. So it's a little bit iterative. And I think that's common in a new space where you need to take a few bites at the apple to really understand it. So that's one. Number two is what I think a lot of pharma will be doing is thinking about what's their process for identifying new targets versus what we bring to the table. And as I described, we could, in silico, through our database, very rapidly frame up key elements of a target that in the past might have taken years to get those data around. So once people understand that, they're like, okay, wow, you're saying you could give me a list of X number of targets that have A, B, and C and D features that I'm looking for. I'm like, yeah, we can do that tomorrow. So that's like very attractive. And then your point about, is it maybe early to invest? And again, I think the Pfizer deal exemplifies why for many it might not be, is that one element is that some of the proteins themselves will be the drug. Think like insulin, GLP-1, GIP. These things are peptides that are administered, and it's not like you've got to create, a lot of new composition of matter. So there's a more rapid route to using the protein as a drug. So that's one element. Look, people are going to be doing drug discovery for some time, and where a lot of the companies are stuck now is they're competing with each other on the same targets. And I knew that really well. Like every target presentation would have a big section on, okay, where's Pfizer, where's Merck, where's Novo, right down the line. And everything we have, like no one is working on it by definition because it's new. Yeah. And that's attractive.
Yeah. Was that part of that? And that's a great segue to the Pfizer deal. You mentioned that everyone's competing on the same targets, and I mean the GLP-1 space is probably a prime example there. So was that kind of key to Pfizer's interest in ProFound and what you're doing, that sort of novel target?
Absolutely, yeah. So the whole proposition is novel target, novel mechanism of action, complementary to GLP-1.
Yeah.
GLP-1s have been fabulous. I mean, it's just amazing. I worked on one at GSK years ago. The percent weight loss and what we're seeing now is pretty phenomenal, but it can't be the only mechanism that could lead to benefits for patients with obesity. So there has to be orthogonal mechanisms. And that one is, acts centrally, but finding proteins that act locally, for example, on fat, is something that, there's enormous opportunity for novelty and to be first in class.
So tell us, I put the cart in front of the horse here, Dr. Lepore. I jumped right into this Pfizer deal and made the GLP-1 reference. And I don't think we framed up the context of this deal. It's in the obesity space.
Yep.
So I'm going to hit rewind and then allow you to take it.
Okay.
where you would normally start with the explanation of the Pfizer deal.
Well, I think we were both complicit in jumping to the end, but we'll conspire to go back to the beginning. Look, let me go back even further. We have we being Flagship pioneering. And by the way, my role is constructed in a very cool way where I'm CEO of ProFound Therapeutics, but I'm also have a role that's called CEO partner in Flagship. And it's intentionally designed that way so that I have visibility and ability to contribute to other things Flagship are doing. So as in wearing my Flagship hat, I think it was about a year ago, Flagship and Pfizer put together a deal where each contributed 50 million. And so there's a 100 million pool that could be applied to drug discovery programs that come out of any part of Flagship. And the idea was, look, Pfizer are just outstanding in terms of drug discovery, drug development capabilities. They're interested in first-in-class medicines. Flagship have all these creative platforms that are generating ideas, molecules, and potentially assets. And then the idea is use this 100 million to fund 10 asset specific programs.
I think you alluded a bit to the differentiator with this early program with Pfizer that you're working on, how it's sort of differentiated from the central application of Ozempic. Can you elaborate on that a little bit? Like, what is it about this specific project that piques Pfizer's interest?
Yeah, look, we haven't disclosed any further details of the science and exactly what we have and what we're looking for. But I could maybe frame it up a little bit more in the sense that the enormous value of our platform is to identify things you couldn't have construed were there. That's the point. They're novel. So, it wouldn't make sense for us to say, well, okay, let's find GLP-1. There's 10 GLP-1s. Yeah. The point is to find novel proteins that regulate the pathogenesis of obesity that could be used, orthogonally with GLP-1.
Yeah.
And it's, our platform enables us to identify potential proteins that could do that. And then we'll have a screening mechanism for identifying them, but we haven't disclosed anything beyond that.
Sure. Yeah. I mean, it's safe for us to assume that it's different and frankly better. I mean, to your point, like I think it wasn't too long ago I read that Hims has come out with a super price competitive GLP-1 offering, like $111 a month or something like this. Right. I mean, that happened quickly, right? Like that space.
Not surprising though. Yeah.
Not surprising at all. Yeah.
But for some, that business model makes sense. If you want, 0.5% of, 50 billion market, you can make math work for a certain company, but that's not what we do. We're here for novel targets and novel biology.
Yeah. Yeah. Very good. Well, what's sort of the next step in that partnership?
Like, the next step is to do the work and identify what we're looking for, move it along, us to get milestones, Pfizer to get great assets, and rinse and repeat.
So there's a whole bunch of next steps.
Yeah.
Yeah. Very good. So we've talked a little bit about sort of some of the hints as to the indications, modalities that you guys might be leaning into. Do you have any other in the works plans that can be shared in terms of sort of the productization of either the platform or its discoveries?
Yeah, look, again, the oncology stuff is really exciting, and we'll be seeing that progress over the next, years, months and years. I talked about immunology, how we're focusing on secreted molecules or receptors for secreted molecules, and the human genetic insights to how to pick those is really exciting. So that's something we'll be really pushing. And then in metabolism beyond obesity, there's really important opportunity to think about molecules that affect muscle, for example, or other organ systems. So that's an area we're working on. And then, the platform's amenable to so many other areas, and we'll be creatively finding ways to build value in those areas too.
Yeah. Very cool. What is the next big box to check for you personally in your roles, both as CEO partner at Flagship and at ProFound?
Yeah, for me, it's I have a short focus on proving that these molecules are going to make medicines, and just like pushing these through. And I don't mean pushing them through no matter what. I mean, like really doing the hard work of finding the ones that have the most promise for patients and most differentiation. And this is like, we kind of have the whatever the expression is, the bull by the horns or something. Now we just have to wrestle it down. And it's going to be an exciting couple of years to see this progress. Because, I joined, as I said at the outset, the vision is that this is not just cool science. Oh, wow, how neat it is that there's more proteins. The box to check for me is that that's all true. And we got something over the hump and it's really in patients and doing something important, that'll be outstanding.
Yeah. Very
And
good.
I'm confident we're going to get there. It's just how
Yeah.
many.
I mean, you know, like we ascertained from the outset of the conversation, you know, that you're coming from a place historically in your career where, you know, you played an active role in the effect of the therapies that you administered. You know, moved on to development of therapies. There's little doubt that you would jump onto a project just for the fun of it at this point in your career, right?
Yeah,
So,
that's right. The Bayesian prior is low on that. Yeah.
yeah. What have I not asked you about the work you're doing there that I should have or would have if I were a better interviewer?
Well, I won't accept the premise about the better interviewer, but what might what have we not covered? Let's put it that way. One thing, I'm just kind of looking out my office window and sort of could see some folks walking around. It's like a really cool place to work. And we've attracted some just amazing talent from like different quarters of the world, literally and figuratively, because to do this, it's quite an interdisciplinary challenge where this computational biology, genetics, proteomics, biology, now even like clinical development. And it's not the standard roles that are in pharma to do this because it's a new kind of science, and the application of technology and computational biology and AI ML to this is like a new way of approaching target selection. So, you know, it's a really cool environment and we've attracted really cool people. And that's been a lot of fun for me to see them starting to nourish their careers and seeing them turn the corner from, oh, this is really cool to, hey, this is really cool and we're making medicines. So that's been fun and it's a cool place to work.
Yeah, I mean, I get that sense as I noted. I've interviewed several flagship CEO partners and CEOs of flagship companies, and the computational angle it's a common thread among, I think, pretty much all of them that I've spent time with, and it has raised the question in my mind that maybe you'd be interested in addressing it, and that's like there are dozens of flagship companies, and computational and AI and ML are integral to so many of their platforms. How do you like how is there differentiation from one to the next within these flagship pioneering companies that are using these computational tools? And is there at any point risk of them all sort of doing the same thing? I know it's a maybe a simple and naive question, but
it's a good question. So a couple things. First off, there's a central group called Pioneering Intelligence, run by a really amazing leader, Armin Merkidjian, who has like it's a central group that's really about nurturing new technologies, new approaches, and being a central place that we can draw from in terms of expertise.
Mm-hmm.
And that's really been great because, you know, we meet with them and we get ideas about, oh, well, with your database, how come you're not doing X and Y? Oh, that's a really cool idea. How do we do it? Well, we did it over here. So maybe you could use that model, but you're applying it in a different way. And then it accelerates our ability to use the methodologies. So that's been great. But no, I think we're all doing computation, but it's actually fine if some of it is applied, if the same methodologies are used. It's the substrate that's different. Mm-hmm. So we have a database of more than 1 trillion data points. It's like ridiculous across like genomics, genetics, proteomics, gene regulation, et cetera, integrated in a single database. And with that size of data, it's really a fabulous substrate for doing like machine learning models that start to predict things. Mm-hmm. But what we're trying to predict is what's the function of this novel protein. Over here in another flagship company, they might use a similar model, but they're predicting something else. So the overlap isn't on the science and the outcome. The overlap is on the methodology, which is great. That's by design.
Yeah. Yeah, very good. That helps a lot. And in a sense, like I get the sense that even though, like you mentioned earlier, when you were at GSK and you had a question or needed some information, like there were a multitude of resources to turn to. This the flagship pioneering ecosystem kind of presents you with
Yes,
the same concept, right? A whole lot of resources. I mean, you can pick up the phone or walk across the hall and
100%. It's one of the reasons I came. I probably left that out at the beginning. Yeah, it's kind of the best of both worlds. We're a company, ProFound Therapeutics, but we're nestled in this ecosystem where we call people and get help, and they're incentivized as well because we're largely owned by Flagship and as are the other ones. And then there's these central groups like Pioneering Intelligence, I mentioned Pioneering Medicine, that are specifically designed to help us be successful. So it really is the best of both worlds in that sense.
Yeah, very good. Well, Dr. Lepore, we're short on time, so I'm going to let you get on with your day. But this has been a fascinating conversation. This is one I say this when I'm like really intrigued by what comes next. I want to have you back on the show, like in 6 months, 8 months, a year down the road. Yeah, just for a catch up and to learn about progress, because it's such an exciting discovery and there's so much to be done with it. So hopefully you'll be agreeable to come back on and join us at a later date.
Absolutely. It's been a lot of fun for me. You can tell I love to talk about. I don't as much love to talk about myself. That's always challenging. I love to talk about the science we're doing and how this would come together. And really appreciate your interest and your listenership's interest, and look forward to connecting.
Awesome. Thanks, Dr. Lepore.
All right. Take care.
So that's ProFound Therapeutics CEO, Dr. John Lepore. I'm Matt Pillar, and you just listened to The Business of Biotech. We are proudly supporting Bioprocess Online's Bio Expo Live, which kicks off very soon on July 30th and runs through August 1st. It's 3 days of virtual programming on all the latest and greatest upstream, downstream, quality, analytical, and data solutions. You can register for any one or more of those sessions for free at the link in the show notes of this episode. Register today. We'll see you there. And in the meantime, thanks for listening.